A-level revision video for biology, and this is Mr. Phillips, and today we'll be looking at mutations. In order to start looking at mutations, I want to have a look at this question. Please feel free to pause this video and have a go at this question if you would like. The question, if you look at it, says the yellow colour of peas is a result of an enzyme that breaks down chlorophyll, which is green. The dominant allele codes for the production of the enzyme that breaks down chlorophyll. The recessive allele is mutation, and it codes for an inactive form of the gene. Outline how the Y allele codes for different production of this enzyme, and explain why the Y allele codes for an enzyme with a different primary structure. So this question is only 6 marks, okay? But if you were given a bigger question on this, because you could quite easily be, how would you talk about it? Well, you would talk about quite a few different things, because you could talk about mutations, you could talk about protein synthesis and transcription and translation, you could talk about the DNA structure, and therefore it shows you how much it actually all links together in the topic that we're about to cover. The actual mark scheme for this question. So you see here they split it into 3 or 4 different sections. You have stuff about the genetic code, of which you talk about the general genetic code and how it works. And obviously that's explaining then how the proteins are going to be made. You then have transcription and translation, and then you have information about substitution, mutations, and the impact of that. And obviously we're going to go through a lot of that today, but just as a show of how varied this area could link to. Diagram which shows you mutations in one diagram. So effectively we have here GAG in the gene sequence. That has had a substitution reaction, so the A has been substituted for a T, and that then has caused, in this case, the egg and sperm to change, which will cause a phenotype change in the offspring as a result. Now, there are different types of mutations. So mutation generally is a change in the amount or arrangement of genetic material in a cell. And there are chromosome mutations and gene mutations. So chromosome mutations we don't really look at A level, but there are 3 sort of different groups. There is changes in the chromosome sets, which often is something called polyploidy. This does happen in plants, and it could be a case that you might have a diploid plant, say instead an egg is diploid instead of haploid, combining with the pollen, which is haploid. So if you get diploid and haploid go together instead of haploid and haploid, you get triploid. Plants are hard enough to withstand that, very unlikely in animals. Changes in chromosome numbers, so this is one we'll talk about in a second. So the example you're probably aware of is Down syndrome. And then also the chromosome structure itself can be changed. So the one that you're expected to know from when you did mitosis is non-disjunction chromosome mutation, and this is where the chromosome may fail to separate properly during metaphase into anaphase, and therefore you get the incorrect haploid number for fertilization. And you'll have more chromosomes than you should have if this survives, and as a result of this, we know that if you have an extra chromosome for pair 21, then it leads to Down syndrome. So there are lots of different types of mutations. You see here a table which shows us this codon TTA. Here it has been changed to TCA. Okay, so just one, the T has been substituted for a C, and that has changed the amino acid it codes for, so it's called a missense mutation. Here, though, the TTA has become TAA, which codes for a stop codon. So then no more amino acids after that point will be coded for. That would obviously have a massive impact on the protein, and that's called a nonsense mutation. We could have a silent mutation where the TTG has replaced TTA, but it codes for the same mutation, same amino acid. Or you could have a frame shift where, in this case, the T has been deleted, it hasn't been replaced. So all the amino acids have shifted one position to the left, changing all the codes have been shifted, all the bases have been shifted one place to the left. So therefore, every amino acid to the right of that mutation will be impacted, causing something called a frame shift. Let's look at them in more. Main groups of mutations we'll be looking at is first substitution mutation. Now I mentioned bases earlier, and obviously it is about the bases changing, but be aware it's not just the base, it is the nucleotide. So the nucleotide is the phosphate, the sugar, and the organic base. So it's the nucleotide in the section of DNA is replaced or substituted by another nucleotide with a different organic base. And we can have a couple of different examples here, and you can see what those examples are. So here we have the original letters. So we've got here, sorry, I should have said, we've got the A, the U, G, U in the DNA, in the RNA sequence, because it's U instead of A. That then gets replaced in this one by UGC. So this original DNA coded for the amino acid cysteine. If you change the letters of UGU to UGC, it is a neutral or silent mutation because it still brings in, even though it's a different codon, the transfer RNA still brings in the same amino acid. So it still codes for cysteine. The second one you can see here is we go to UGG, and this one is a missense mutation because it changes the codon and it brings in a new amino acid, tryptophan. And then we have a nonsense mutation, which we talked about a few minutes ago, which is UGA. And at UGA, that would bring in a stop codon, and a stop codon basically means no more tRNA is going to be attached. So it's going to have a bigger effect because none of the following amino acids are going to attach either. Substitution mutation is what effectively causes something called a point mutation, and this is when only one triplet is affected and there's only one amino acid in the sequence is changed. Although obviously for a nonsense mutation, none of the next amino acids in the sequence are going to be able to attach as a result. Substitutions lead to point mutations, and that doesn't mean they're only a small effect. It depends on obviously the actual mutation that's happened. So a big example of a very big damaging point mutation is sickle cell anemia, which is where one base or one nucleotide, I should say, substitutes at codon 6, and this alters the polypeptides in the beta chain of hemoglobin. It makes the amino acid 6 get replaced from glutamic acid to valine. But the importance is that glutamic acid is hydrophilic, valine is hydrophobic. So it impacts the amino acids, and it makes the hemoglobin protein change, and it causes your red blood cells to go from the shape you know to sort of a sickle shape, which sort of looks like that. And that may render the red blood cells useless. They carry less oxygen, but also in low oxygen concentrations, you end up with something called a sickle cell crisis, which is where they collapse and they can block up your capillaries. So if we keep looking, then there are different mutation types though. So we've looked at substitution. You can also have deletion, where the base actually gets deleted. So here you can see we have G and C, and then C and G there, but that has been deleted. Okay, so we go straight there. That would cause a frame shift. Or you can have insertion, where you add an extra nucleotide, and therefore it adds it changes all the amino acids after that point. This is called a frame shift, and it basically means it moves across as a result. If you have an insertion, if we have a look here, we have the original one at the top, so it goes T, C, A, T, and you see the amino acids here. So there was the T that's still there, but instead of the C being next, they've inserted A, and therefore every amino acid after that point is going to be different because you've altered the codons, moved them along, shifted them along by one. So what was originally CAT has now become ACA. What was originally CAT has become TCA. So it's caused the frame shift of all the subsequent codons following the mutation. And here we have a deletion. So you can see here the letter A has been deleted, and again that's caused a frame shift now. So instead of CAT, it becomes CTC, and again all the amino acids following the mutation will be. So to compare frame shifts to point mutations, to have a look at this, there is only one base pair being replaced in the point mutation by substitution, which we know is insertion or deletion for frame shift. It occurs only in one single nucleotide for point mutation, whereas multiple nucleotide alterations can occur for frame shifts. The sequence of nucleotide is not altered, but it is afterwards, after the mutation. It only occurs in this, only substitution of the base occurs in this mutation, but insertion and deletion occur for this one. The structure of the gene changes, the number of nucleotides in the gene changes for that one. There are two types you don't need to really know about that one, and sickle cell anemia is an example, and Tay-Sachs disease is an example of a frame shift mutation. So just some other types of mutations then. So there's a duplication mutation, and this is when one or more bases in the DNA code become repeated. So the BC there becomes repeated, and this will obviously produce. You can get an inversion, which is when a group of nucleotides become separated from the DNA sequence and then are rejoined, but they're reversed around. And this obviously causes a different sequence of amino acids. So GCA codes for alanine. Inversion would lead to ACG, which codes for free. Translocation of bases, which is where some of the bases might move along from one segment, one chromosome to another one. Okay, so groups of bases become separated, and then they might be inserted into a different chromosome. That would obviously have a significant impact on gene expression, and they think this can happen to lead to increased cancer chances. Now, so how do mutations happen? Well, we know mutations are random events, but we also know that there is a low chance of happening. So we can increase the risk by being exposed to mutagens. There are two types of mutations. There are spontaneous mutations, which are permanent changes that occur without any outside influence. Okay, and obviously there is relatively predictable frequency. So the natural mutation rate is about 1 to 2 per 100,000 genes every generation, but it can obviously be increased. Two types of mutagenic agents are high energy ionizing radiation that have enough energy to change the chemicals in DNA, and chemical substances which can cause and interfere with transcription, protein synthesis. Mutations don't always have a bad effect, so we said it might be a neutral effect, and that might be because it occurs in a non-coding intron region of the DNA. It might be because it substitutes but for a new codon that codes the same amino acid, or it might be that there's no selective advantage. So some people really hate the smell of honeysuckle plants, and that doesn't seem to have any advantage. Some people can taste things differently, so you might be someone that can't stand the taste of sweetness put in like Pepsi and Coke to make the sugar amount less, and some people don't even notice the taste, and stuff like tongue rolling doesn't have a specified the environment itself massively determines if a mutation has an advantage. So skin color, you can see on this map where skin color naturally evolved, and just depending on where you are on the planet would depend on whether your skin color is an advantage for you in terms of survival. So if you have darker skin, it means you've got more melanin in your skin, and that means you would have a greater survival chance if we didn't use stuff like sun cream, etc., around the equator, because melanin prevents you from absorbing as much UV, which reduces your risk of skin cancer. If you were very pale and you lived on the equator with no protection, you would have an increased risk of skin cancer. But if you were living in the Siberia, let's say, then dark skin would mean you wouldn't absorb as much sunlight, and therefore your UV, and you'd have less vitamin D. Too much because we put sun cream on, we can take tablets and alter our diet to make sure we've got vitamin D. But it shows you that the environment does determine whether a mutation is beneficial or a disadvantage for survival of an organism.